All right, so insert page. So we're going to build a picture here. I'm going to draw it on this board this time so that those people at home can see it. But we started out with a pyruvate, turned it into a acetyl-CoA, ultimately added 4 carbons to make 6 carbons. That's how far we got. So I need to draw those things real quick so you can kind of recap and think about what's happening to start. So I got a pyruvate here that is going to, well, it has 3 carbons currently. It's going to lose a carbon, and it loses that carbon as what, guys? Every time you lose a carbon, it turns into what? Carbon dioxide. There you go. So I got a CO2 that gets kicked out, but I also made an energy molecule, that NADH, from an NAD+. And then that 2 carbon molecule just turned into this acetyl-CoA. Now remember, all that means is that I attached an enzyme that's called CoA, coenzyme A, to that 2 carbon molecule. This still has 2 carbons just like it did before. So this still has 2 carbons. It just has a fancy name now. All you need to worry about is how many carbons are in there right now. But I was going to take this 2 carbons, and I was going to add it on to 4 carbons to make 6 carbons total, and that's my citric acid. So I know we got at least that far in the picture. Is that it?
Mm-hmm. Mm-hmm.
Nope, you completed the loop down here. This coenzyme A then is released, and so it's separate again, and it can go back and find another 2 carbon molecule to add on to another 4 carbon molecule. So I actually have a little mini cycle right here. This coenzyme keeps grabbing more 2 carbon molecules to add on to more 4 carbon molecules to make more 6 carbon molecules. That's citric acid. So that actually catches us up to where we were supposed to be, correct? Got everything up there now? We did talk about citric acid a little bit, maybe. You guys should be recognize that. What do you find it in?
Like fruits.
Citrus fruits, right? Like oranges and lemons and limes. And it is actually very sour. That's what they concentrate to put on a warhead, right? So it's very sour. Some people hate those things. Some people love them, right? It kind of depends on the person because everybody senses their world differently. So sour is unbearable for some people like it, right? So citric acid is something you're familiar with. That's also why this is called the citric acid cycle sometimes, because it's part of the cycle. It's a molecule that's in the cycle. But from here, right, we need to loop up, come back down, right? I'm actually going to be crunched over here for space, but I'll figure out how to do it somehow. It doesn't really matter. Just make sure that you try and draw your picture appropriately too, because Kay's going to be very scrunched for space on his too. Right? You need a lot of room going this way. Right? But we'll see what happens. If you got to start all over again, you just draw a new picture. It's not a big deal. Right now, you'd actually have a lot of time to catch up if you wanted to start all over again. You should still have a lot of room going this way. Right? Yeah, you'll probably. So whatever happens, make sure you have a good picture when you're done. If you got to start all over again, it doesn't matter. It's not like it's going to take you that long. Plus, you could open this video later on Google Classroom and you could just draw whatever's there. It might take you all of 2 minutes to draw another picture. It won't be that big of a deal. So citric acid, next, is going to turn into a 5 carbon molecule, right? And what you guys are supposed to recognize and see a pattern with is just like in that very first step. I lost a carbon here. I went from 6 to 5. Just like over here, I went from 3 to 2 in that very first step. And every time you do that, you eject this same molecule. And if you lose a carbon, you always lose it as the same thing. So the only thing missing in my picture right now that needs to be kicked out is what, Kate?
CO2.
A carbon dioxide. So between here and here, I lost a carbon, so I'm going to kick out a carbon dioxide. Every time you lose a carbon, you're always going to do that. And that is that carbon dioxide that you are currently exhaling, right? You are expelling that into your environment because you build up a lot of that stuff inside of your body and you got to get rid of it. It's toxic. But now that I'm at 5 carbons, the other thing that I should recognize here is I did get smaller. That is going to be exothermic. It is going to be catabolic. I should catch a little energy again. So I'm going to catch some more energy in another one of those NADH molecules from an NAD+. Right? Similar to over here, right? I lose a carbon, it's exothermic. I should definitely get some energy out of that and catch that energy with an anabolic reaction. Right? So that actually covers all the way through here 4. We're just building off of that. So you can follow along either way with your notes, your salmon colored notes, or with your picture you're drawing. All the information's in both places. But the next step, again, I'm going to go from 5 carbons down to, I want to make that arrow longer, so I'm going to fix that actually. I'm going to go from 5 carbons down to 4 carbons. So if you've been following along at home, right, some people literally, if they're watching this video, I lost another carbon. So I should kick out another molecule. What molecule needs to be kicked out right now? Coal. They got kicked out 2 times already. Every time you lose a carbon, you lose it as what? Carbon dioxide. So I'm just going to kick another one out. Again, I throw those off to the side instead of putting them in the cycle because they're not part of the cycle, right? Those things are waste products that need to get out of your body eventually, right? So we don't keep them in the cycle. We don't even try to keep those inside of your cells. You got to get rid of that stuff. But now I'm down to 4 carbons. This step is also catabolic, right? Energy is being released. I'm going to catch that energy. This time I get to make, let's check real quick because usually I have this available to see because I don't remember all this perfectly, right? Sometimes I have to refer to the notes a little bit. This time I'm actually going to make 2 energy molecules. I'm going to make an NADH again, which I'm going to go off, right? I'm going to go this way into the middle to make that. But again, apparently there's a lot of energy being released there because I get to make 1 more energy molecule. And this one is going to be an ATP. Hopefully that didn't go off the screen too far because I'm recording so much of this screen, but it should be all right. So I'm going to make an NADH and an ATP here. Now that's important because you guys got to remember that ATP is that energy molecule you can use for anything. That's actually good to go right now. That's energy that your cell can use for whatever you want it to be used for. This energy though in NADH is still trapped. I have to take this one step further to get that energy to be useful. But that ATP should be good right now. Final step of the process, I need to change this 4 carbon molecule into that 4 carbon molecule. And they look the same right now, right? So what we're going to do is put a, can never think of that word. We're going to put an apostrophe by one of these. Just mark it so that it looks different than the other one. So I think the notes have that one. Let me check real quick. Yeah, I made the product have the apostrophe. So this 4 carbon molecule is different than that 4 carbon molecule, right? Make sure you can see that. But also, this 4 carbon molecule has more energy than this 4 carbon molecule. Again, this is going to be catabolic, so I need to catch energy one last time. And I'm going to catch energy in 2 products here. I'm going to make an NADH one more time. But then I'm also going to make something new, something you haven't seen before. It's actually similar to that NADH. It's called a FADH2, right? So this FAD is actually very similar to that NAD. The only difference is that it came from something that had a +2 charge to begin with, and I'm going to add 2 hydrogens to it, right? So if you remember back from photosynthesis, that NADP plus, right, you added a hydrogen to it, and it caught 2 electrons to make a bond between that hydrogen and that molecule, right? So the same thing's happening in NAD plus. You add 2 electrons plus a hydrogen ion. You can make a bond between NAD and a hydrogen. But here, since it's a +2, I'm going to add 2 hydrogens. And that means I'm actually going to catch 4 electrons. So this is a high energy molecule, but it has a couple extra electrons compared to the NADH. But otherwise, you don't really need to worry about that too much. You just need to know that it's high energy, that it holds some electrons that we're going to use later. Make sense to everybody? So this actually is a complete picture now, because now this 4-carbon molecule, you could add another 2 carbons to it, make it 6 carbons again, and start all over, right? So this is my cycle over here on the right. So it just keeps going round and round and round. Over here is kind of a lead-in to my cycle, because I have to mess with that pyruvate before I can add it to the 4-carbon molecule to make it 6, right? So it's a little bit more complicated than the Calvin cycle that we did last section for photosynthesis. There's a few extra steps, but actually it's pretty similar, right? In the Calvin cycle, you are bringing in carbon dioxides, correct? And you are making a 3-carbon molecule. Here you start with a 3-carbon molecule, you kick out carbon dioxides as you go. Just the opposite, right? And in the Calvin cycle for photosynthesis, you are absorbing energy, trying to make a high energy product. Here you're releasing a bunch of energy with catabolic reactions. I'm going to use that energy that's in these energy molecules that I caught later for electron transport. So everything's backwards. It should actually feel a little bit familiar. It's just in the opposite direction of what we did before. So you need a picture like this. Next thing we actually have to do, though, is balance this whole thing. Because how many pyruvates do you get out of a glucose? If I go backwards, when I broke glucose in half,
2.
how many pyruvates did I get? I got 2 of them, right? So that goes back to another thing we were talking about before. Remember when we did a 2 in a different color, right?
Yeah.
Same thing here. I'm going to put 2s all over the place, but they're going to be a slightly different color. There are a ton of highlighters in those beakers over there. If you want to go grab a couple colors, that'd be a good idea. You could even grab all 4 colors. There's pink, orange, yellow, green, red. Get what you need. Because I'm going to highlight a bunch of stuff on this slide, and you might need that stuff.
My entire head hurts just opening my mouth.
Just hit that. Yeah, we're good. So, I'm going to add some 2s, but you're going to do them a little bit different. If you could highlight them somehow red, or just use the highlighter to make the 2, I don't really care. But I actually had 2 pyruvates to start. So every step along the way, right, instead of having 1 product, I have 2 products, right? So I'm actually, from 2 different pyruvates, going to kick out 2 different carbon dioxides, right? I'm going to have 2 of these 2 carbon molecules. I'm even going to make 2 of these NADH molecules, right? Each time I make 1, but for 2 per glucose, I'm going to make 2 of everything, right? So that means I'm going to have 2 of these acetyl-CoAs down here. That means that I'm going to do this little entry twice to add 2 carbon molecules to 4 carbon molecules, but I'm going to have 2 2-carbon molecules, 2 4-carbon molecules to make 2 of these citric acid molecules. And then I'm going to drop from here to here, losing 2 carbon dioxides in the process, making 2 NADHs in the process, right? Should actually start making sense here. Everything's just a 2
Okay.
as I keep going, right? So from here, 2 of these turn into 2 of these, which means I kicked out 2 carbon dioxides instead of 1. I'm making 2 NADHs. I'm making 2 ATPs. I'm going from here to here twice. So I don't just make 1 NADH, I make 2 of them, and I make 2 FADH2s. So just like in the Calvin cycle, how many times do you do this per glucose?
2.
Twice, right? So the whole thing has to happen twice per glucose, just like in Calvin cycle, I had to do it twice to get the carbons I needed to make the glucose. We're doing everything the same, just in reverse. So we had a ×2 that we were supposed to understand what that meant in our Calvin cycle picture. We got 2's all over the place again here. Because there's 2 pyruvates per glucose, you get to do this whole thing twice. But then I go through and start looking at my molecules that I made. And I need to keep track of those things, right? Some of those molecules are good, some bad, right? So again, you can highlight these some color if you wanted to. How many, as I go through, carbon dioxides are being produced overall?
3.
6 of them.
Oh, yeah, 2.
Because each one's 2,
times 3 is
right?
6, yeah.
So I got 6 carbon dioxides. How many carbons does glucose have in it again?
6.
6. So that makes sense, right?
Yeah.
I'll just kick out 6 because I brought in 6 from the very beginning. So that's all balance, conservation of mass, right? I'm catching energy molecules along the way, right? How many of these guys am I making overall? I gotta search for those. But there. How many of those did I make? Made 8 of them, right?
Yeah.
So I got 2 here, 2 there, 2 there, 2 there. So I got 8 NADH molecules. But if you think about it, I made 2 over here too, right? So that's how many total now? 10 NADHs. So again, just kind of keeping track of things. I had 6 carbon dioxides. But that makes sense because glucose has 6 carbons in it. If I go back here, glucose has 6 carbons, right?
Mm-hmm.
So I should be kicking out 6 carbon dioxides. But then I also made 10 NADHs. I'm including the 2 that were made in glycolysis, plus 8 more here, but I got 10 total. That's on your notes, you might want that, right? I'm putting that here. You could also put that near that in your notes, in your orange, salmon color notes, whatever. I also made some other energy molecules. This one, right, was really important to us. But how many did I make?
1. 2.
I made 2. Now, again, we talked about that before. I already had a net gain of 2 after glycolysis, but then I lost 2 to move pyruvates into the mitochondria. Now I'm getting 2 back. So I'm still at a net gain of 2, right? So I'm not going to add any extra ATP on from glycolysis this time because I actually lost that ATP. So my net gain at this point for ATP is 2. And again, if you're trying to use colors on your pictures, you might color code these things. If you were highlighting these pink, you would highlight this pink, right? Whatever color you were using, if you wanted to keep track, that'd be a good idea. And if you used a different color than me, because it looked like Robert used green for the carbon dioxide, that's fine. Just trying to use different colors so we can keep track of stuff. So one more thing, and it's similar to that NADH, so I'm going to highlight it the same color, but how many of those FADH2s did I make? Made 2 of those. So I'll list those as well up here. I got 2 FADH2s. Right, remember the carbon dioxides are toxic. I gotta get rid of those. You're breathing them out right now. These NADH and FADH2 molecules, there's 12 of those kinds of molecules total. Those are going to be used later to get a lot more energy. ATP, useful right now. I could do whatever I want with it, but I'm still at a net gain of 2, right? Just like I was after glycolysis. So considering I'm still just at 2 ATP, and I can't do anything more if I don't have oxygen. There's really no point in doing this whole complicated process without gaining anything out of it. All right, so I told you that this technically is anaerobic because there's no oxygen in here. But you wouldn't do this without oxygen, even though you could do it without oxygen, because you just make a lot more products that are still useless without oxygen, and you get the same amount of ATP overall, right? These two things are only going to be useful to us if we have oxygen available. So you made a bunch of them, but you didn't really need to, right? So that should actually be a complete picture for your Krebs cycle. We just got one more picture to go. We got an electron transport. And then we have a few questions to answer about that. So hopefully you got a pretty good picture going there. We're going to do one more picture. If you need to flip the page to draw it on the back side, that might be a good thing. If you got lots of room on the front, you could draw it on there, but just make sure that you can have good, accurate pictures for everything. So we're going to do this electron transport next. This is actually a pretty easy picture because there's not that many steps. It's pretty simple. And it's also very similar to what we did in photosynthesis. It's just backwards again. It's not the same direction, but pretty much the same ideas, same concepts. So, answer another page here. And I'm going to start drawing something new. So, in your electron transport, the idea here is that I have a membrane again. So I'll put some phospholipids in here just to start building that so it looks like a membrane. But if I go backwards, the only membrane I'm really worried about, I'm worried about that inner mitochondrial membrane. This membrane right here, right? So that's the membrane where this electron transport is going to occur. That cycle we just did happened in the matrix, which was inside of that inner mitochondrial membrane, right? But we're on that membrane right now doing this electron transport. So what I need in here next for my picture is a chain of proteins just making its way down. So this should look familiar because this looks just like that photosynthesis picture. The only difference is I don't see any green because what's missing?
The one thing, chloroplast.
The chlorophyll, right, is missing. Right, I still have a chain of proteins, but I need electrons to travel down that chain of proteins. And I get electrons from those energy molecules we've been making. Remember I have, how many, was it 12 NADHs? Or was it 10? I can't remember. It's 10 total?
Yeah.
I bet. It's 12 total molecules. But 10 NADH molecules, plus I also have those 2 FADH2 molecules that I've made so far. All of those contain electrons that have high energy. And I'm going to use those to start this electron transport. So they are going to turn back into NAD pluses and FAD plus 2 molecules. That 2 look goofy, so I'm going to do that again. So I'm going to take these things, turn them back into what they started as. But all those electrons that were in those molecules are going to enter this electron transport. So I'm dumping out electrons from here. And those electrons need to travel down this chain of proteins. And just like our picture from photosynthesis, those electrons have energy, right? As they travel down this chain of proteins, I can use that energy for something. And I'm going to use them for exactly the same thing I did in photosynthesis. What did I power in photosynthesis with those electrons?
The
Protein pumps.
Yeah,
I powered protein pumps that were pushing hydrogen ions, right?
Yep.
So I'm going to do the same thing here. I have hydrogen ions that can be pushed across the membrane. And the energy that's helping me do that comes from those electrons. So I'm going to push these hydrogen ions across the membrane, building a concentration gradient on one side, right? So as I keep pushing those hydrogen ions across the membrane with more and more electrons traveling down that transport chain, this concentration continually gets smaller and this one keeps getting bigger, right? So I create this concentration gradient. I create what they call a chemiosmotic gradient, but that's a fancy word you don't need to know. But then two more things need to be in this picture. I have a problem. These electrons can only travel so far, right? I need something to catch electrons at the end of this electron transport. If I don't have something to catch electrons, then the electrons just end up in a traffic jam, essentially, right? They want to go, but they don't have anywhere to go, right? So you just get a bunch of them stacked up on top of each other, and they don't have anywhere to go. So that current that we talked about that's supposed to power pumps that push hydrogen ion across the membrane quits, it stops, right? You can only make this electron transport happen if there's somewhere for those electrons to go. You need something at the end of this transport chain to collect those electrons. And that's why this process is aerobic, right? What do I need to catch these electrons? What would make this aerobic? What molecule's missing that you're breathing in right now that you need?
Air.
Oxygen, right? So that's where the oxygen comes into play. This is aerobic because I need oxygen to collect those electrons. And this is exactly, well, it's pretty much the same picture as last time, just backwards, right? Last time at the end of the two electron transport chains, you ended up with a high energy molecule that caught those electrons. It was called a NADPH, right? So it's similar to one of these things. But where did the electrons come from in photosynthesis? What molecule did they come out of? What was split to make that oxygen and to get electrons? Water was. So when this oxygen collects electrons here, it's going to turn into what molecule in your body? Water. So that's that end game of my electron transport here. I have to catch electrons with this oxygen, but in the process, I'm going to make water. Remember that oxygen you breathe in does not turn into carbon dioxide. It turns into water that's still inside of your body, inside of your cells. It's not enough water that you can quit drinking water, but it does actually supplement that water that you need to be alive anyway. So this has to be here because otherwise there's a traffic jam, right? Those electrons need somewhere to go. If you don't have oxygens, none of this works, right? Just like that electron transport in photosynthesis would not work if there was no water, right? If there was no water to split to get electrons to begin with. This will not work if there's no oxygen to collect those electrons, to catch them at the end, right? So it's like I got this big slide here, and I got a bunch of kids on it, and that kid at the end just sat on the end and didn't get off, right? No fun anymore because we're all stacked up on this slide and nobody can go anymore because we're all stuck, right? So this needs to be a continuous current to push those hydrogen ions across the membrane. It's not going to be continuous if you don't have oxygen to catch those electrons. So we're all good with that? Makes sense? One last step. I need to use this concentration gradient to my advantage. And this is going to be exactly the same as your photosynthesis picture. The last thing you need to add to this membrane is a channel protein over here, or wherever you can fit it in your picture. That channel protein just lets that hydrogen ion come back through because it wants to move through the membrane anyway. And that channel protein is still the exact same enzyme that we talked about in photosynthesis. This is still ATP synthase. It's actually the exact same enzyme that's used by those plants. So as the hydrogen ion goes through, what am I going to make lots of?
Wow. Oh, energy.
It's called ATP synthase. What's it going to make a lot of? It's going to make a bunch of ATP. And if you remember from our last overview section, I make a lot of ATP here, right? Per glucose, this part of the process, this electron transport, makes 34 ATP, right? Which again was the entire point of burning the glucose to begin with. I need this useful energy that my cell can use anywhere to do whatever it wants to do. And most of that ATP comes from this step. I make those from ADPs. Obviously, I would need 34 ADPs to make 34 ATP, but I assume you could pick that up from the picture itself. But now I have 34 plus, if I go backwards, these two, how many total for a eukaryote? 36 ATP. Whereas a prokaryote, they're lucky. They don't have to spend ATP moving. They don't have to spend ATP moving something into something else that doesn't exist, right? So they actually get 2 more ATP than we do. They get the 38 total because you'd have the 2 net ATP down here plus 2 plus 34 to get 38 total. But they do it way, way slower than we do. So it's actually worth it for us to go ahead and do the process with a mitochondria as opposed to without. So I'm making lots of ATP at the end there, but this would not work without oxygen. You need oxygen to make this work. If you don't have oxygen, what happens in here?
It'll just float off and then get a traffic jam.
There's a traffic jam, right?
Yeah,
So the electrons want to keep moving, but they can't because the electron in front of them is not moving, right? If the electron here at the end can't jump onto a oxygen to make a water molecule, then none of the rest of them can move either. I got those electrons from these high energy molecules here. Those are the same high energy molecules that we made 2 slides ago, and then even a couple slides back, these ones right here. So all of that energy ultimately comes from the glucose in the beginning, but there's lots of steps involved in that, right? It's not just one simple step. There's lots of individual steps that occur. This has a ton of steps in it. This has multiple steps in it, right? This one should be really easy for you guys right now because it looks very similar to photosynthesis, right? You pump hydrogen ions across, you bring them back through, make a bunch of ATP. The only difference is where did the electrons come from and where do they end up? That's backwards, but everything else is pretty similar. So we covered 1, 2, 3, 4 here. We made some water at the end. We made a bunch of ATP. That hydrogen, those proton pumps are powered by electrons, right? The electrons come from NADH2 and FADH2. So again, products of cellular respiration. We get carbon dioxide. How many carbon dioxide are kicked out total? So just like before, actually, just like before, there's 6 carbon dioxides that are kicked out. I'm also going to make some water. That water was made from what? What that you're breathing in turns into that water. Oxygen. You have a net gain of up to 38 ATP, right? But that's only for a prokaryote. How many did you get? 36, right? So you could get 2 from glycolysis, 2 from Krebs cycle, and then 34 more from the electron transport. But you just have to remember that you will get these 2, but then lose those 2 trying to move pyruvates into the mitochondria. You do get a net gain of 36 from those 2 things, but only prokaryotes get to keep that 2. Okay? So it's different for prokaryotes than eukaryotes, but you could get up to 38 ATP per glucose. You only get 36. because you don't get to keep that one. Those two. Okay, so just a couple questions left. How much time do we got, guys? 5 minutes or so?
Man, time went by fast.
I know, just like always. I think that's probably